Good afternoon. My name is Silas Norman, and on behalf of the American Kidney Fund, let me welcome you this afternoon to our webinar on APOL1-mediated kidney disease, overview and implications for care. We appreciate you taking the time to join us today, and we expect that we'll have a nice hour of very interesting educational content. Before we begin with our formal program, I want to take time to thank our sponsor, Vertex Pharmaceuticals, and I want to give an opportunity for our representative from Vertex, Dr. Cremisi, to step on and say a few words.
Thank you very much, Silas. Good afternoon. My name is Henry Cremisi, and I'm a nephrologist as well as the Global Medical Lead and Senior Medical Director for Vertex's investigational APOL1-mediated kidney disease program. Vertex is a Boston-based biotechnology company with a record of approved therapies in cystic fibrosis. But Vertex also has an ambitious mission to further invest in innovative science with the aim of creating transformative medicines for other serious diseases, including kidney disease. Our CEO, Reshma Kewalramani, is a nephrologist, and Vertex has advanced an investigational therapy into pivotal development aimed at treating the underlying cause of APOL1-mediated kidney disease, also known as AMKD. From today's panelists, you'll learn about the discovery of APOL1 risk variants and their connection to kidney disease in people of sub-Saharan African ancestry. But even while the understanding of AMKD grows in the clinical community, many remain unaware. Vertex is so pleased to work with the AKF to help raise awareness of AMKD. On behalf of Vertex, thank you to today's panelists, thank you to the AKF, and thank you all for joining.
And thank you, Henry, and thank you again for all the support and partnership with Vertex and the AKF. As we move to our presentation today, I'm pleased to announce our speaker for today, Dr. Mona Doshi. Dr. Doshi is a professor of medicine, is a transplant nephrologist at the University of Michigan, and one of the medical directors for the Kidney and Pancreas program at the University of Michigan. In addition, relevant to today, Dr. Doshi is one of the investigators for the APOLLO study. The APOLLO study is a study looking for APOL1 risk variants in kidney donors as well as kidney recipients. And we look forward to hearing this conversation today and the information you're bringing to us today. Dr. Doshi, welcome.
Thank you, Silas, and thank you for AKF for inviting me, and thank you to Vertex for supporting this. And so let's jumpstart. Next slide, please. So what I would like to cover today for the audience is start out with discovery of APOL1 and its association with kidney disease, then review impact of APOL1 genotype on progression of kidney disease. This is, we are talking the native kidneys. Thereafter, delve into transplant. And lastly, end with some review of ongoing studies, including treatment options. Next slide, please. So starting out with the background, as we all know, African Americans constitute 13% of general population, as you see in this circular ring on the, the red portion reflects the proportion of African Americans in general population. Whereas in contrast to general population, the proportion of Black individuals on the kidney transplant wait list and those with kidney failure is tripled. As opposed to 13% in general population, Black constitute almost 35% of patients with kidney disease. And therefore, as you see here, is Black individuals are disproportionately affected with kidney disease. Next. So a major discovery in 2010 was made as to why Black individuals are predisposed to development of kidney disease. It could be a combination of, socio-demographic factors, but as well as it is now shown that it may be due to certain high kidney risk variants, particularly associated with APOL1 risk variants that are. So next slide, please. So let me delve a little bit deeper into what is APOL1. APOL1 is a gene that we know that, and genes code for medical information in our body. And APOL1 gene is present on chromosome number 22, as you see here. Normally, folks have no risk variants or APOL1 gene. When not mutated, the individuals carrying no risk alleles succumb to infection caused by trypanosomal zomiasis, as you see the parasite down below. What that parasite does is it causes sleeping sickness, and the person gets gravely sick and dies due to infection. As a process of evolution to protect the human race from this sleeping sickness, APOL1 gene evolved into, there was a mutant variant that developed. So individuals carrying one risk allele, as you see in the middle panel, can beat off or can ward off from infection from trypanosomal. As a result, do not succumb to sleeping sickness and can survive the infection. However, individuals on the extreme right-hand side, if you see there, individuals with two risk variants are able to fight off the trypanosomal infection. However, unfortunately, as a result of this two risk variants, they are at an increased risk of developing kidney disease. So let's take a further deeper dive as to how does APOL1 mutation protect against trypanosomal infection. Next slide, please. So as you see, this picture shows you a nice, parasite, trypanosomal parasite. And what the APOL1 does, it helps to cause lysosomal swelling. So swelling of an organelle within the trypanosomal. As a result, the parasite, swells up and it lyses off and dies off. And therefore, the human being survives this very serious infection from trypanosomal. Next slide, please. So how does one get APOL1 risk variants? Like any other gene, each copy of APOL1 risk variants is inherited from each parent, one from mom and one from dad. APOL1 associated kidney disease is an autosomal recessive pattern of inheritance. That means you need both mutant copies to have a chance of developing disease. APOL1-mediated disease does not have 100% penetrance. It does not follow Mendelian pattern of inheritance. That means every individual carrying two copies of risk variants does not develop kidney disease. Individuals carrying two copies need a second hit to manifest kidney disease. Currently, it is estimated that only 20% of individuals carrying two APOL1 risk variants develop kidney disease. Possible second hits are proposed to be either a viral infection or maybe environmental risk factors, and we'll delve into that a little bit later. Next slide, please. So individuals carrying two APOL1 risk variants or two copies of the mutated APOL1, and only few of them, as mentioned before, only 20% of these individuals develop this classic feature called focal segmental glomerular sclerosis. So as you all know, kidneys help to filter blood. So each kidney is composed of over 1 million tiny filters, as you see in these circles in the middle of your slides, right? These circles normally help to filter the blood. When these filters get clogged up or injured, you see a lot of dark pink, as you see there, varying degrees of dark pink. That is called global sclerosis. So individuals who have two risk variants and have a second hit, either associated with HIV infection on the extreme right panel, or perhaps due to any other currently unknown environmental risk factors, are predisposed to developing focal glomerular sclerosis. And as you see, the risk due to carrying APOL1 risk variants can vary anywhere from 7-fold to as high as 30-fold in individuals carrying with HIV infection. Next slide, please. So there have been several studies since the seminal finding in 2010 showing that there could be a linkage between APOL1 variants and kidney disease. There have been several case control studies, as seen in the blue lines there, several CKD cohort studies, and population-based studies showing a link between APOL1 and kidney disease. Next slide, please. Next slide. So, as mentioned before, APOL1 risk variants were developed in response to trypanosomal infection. As you see, trypanosomal infection was quite common in West Africa, as seen by the magenta dot, which shows you the frequency of APOL1 risk variants. The magenta dots are the areas where the risk variance is at the highest frequency, which correlates with prevalence of trypanosomal infection. So as you see, not all portions of Africa, the individuals were affected with trypanosomal infection. However, trypanosomal infection was quite common only in West Africa or Sub-Saharan Africa. And then individuals who migrated, either due to slavery or otherwise, to other areas of the world carried that APOL1 risk variants with them. So as a result, what you see here is Africans residing in America, Africans from Brazil, Afro-Caribbeans are also carry high-risk variants. Next slide, please. So what is the prevalence of 2 APOL1 risk variants? As I mentioned before, each person inherits one copy of APOL1 gene from each parent, and the inheritance is autosomal recessive. In general population, around 14 to 15% of individuals carry 2 APOL1 risk variants. Next paragraph, next slide, please. So, what are the proposed theories for APOL1-mediated kidney injury? As mentioned before, not every individual carrying 2 APOL1 risk variants develop kidney disease. A second hit is required for an individual to develop kidney disease, which could be either hypertensive associated kidney disease, where the injury is at the level of blood vessel, or individuals could manifest FSGS, where the injury is at the level of the filter, where the blood is cleaned in the kidney, or due to accelerated formation of HIV-associated nephropathy. Currently, the proposed second hits are either viral infection, such as HIV, or recently, we also saw individuals with 2 APOL1 risk variants who contracted COVID infection were more likely to develop FSGS. So viral infections such as HIV and COVID infection are proposed to be second hits. Other proposed hits are environmental hits such as pollution, or perhaps there is a gene-to-gene interaction and there might be an interaction with sickle cell disease. At the cellular level, at the level of the cells in the kidneys, how does APOL1 cause kidney disease? Either it affects the mitochondria or it affects the cells that line these filters called podocyte. And as a result, there is an accelerated loss for podocyte, causing damage to the filter, and a result, a lot of proteins spill out into the urine and the patient gets kidney disease. Next slide, please. So as mentioned before, APOL1 associated kidney disease is a spectrum of disease. In some individuals, it manifests as FSGS, where the filter is plugged up with that dark pink material, as I showed you before. In some individuals, particularly those who are HIV positive, it causes HIV-associated kidney disease. In some individuals, it causes hypertension. It looks like they only have hypertension and have accelerated hypertension-induced kidney disease. It also accelerates development of lupus nephritis, or other non-diabetic kidney disease, and even in certain cases, diabetic kidney disease. So as you see, APOL1 nephropathy is a spectrum of disease, and the impact of APOL1 risk variance on a particular type of kidney disease varies, as you see by this light blue conic cone in the background. The highest association is with HIV nephropathy, and the lowest or no association is with IgA nephropathy. Next slide, please. So let's delve into what is the impact of APOL1 genotype on progression of kidney disease. So in this seminal paper published by Parsa et al., he looked at a cohort of individuals that was enrolled in the AST trial. AST trial consisted of Black individuals, and who had hypertensive-induced kidney disease, and they studied the effect of blood pressure controlled on kidney outcomes. What Dr. Parsa and his group did is they genotyped all the Black participants. AST cohort actually only consisted of Black participants. All the Black participants were genotyped for APOL1 risk variance. So if you focus your attention on panel A, you see the progression to primary outcomes, which is doubling of serum creatinine or starting dialysis on individuals carrying 0 or 1 copy of APOL1 risk variance, shown in the top with the dashed lines, and the rates of doubling serum creatinine or developing end-stage kidney disease in individuals carrying 2 risk alleles. What you see is individuals carrying 2 risk alleles are more likely to double their serum creatinine, that means experience very severe degree of kidney failure, or even reach end-stage kidney disease. So in other words, presence of APOL1 risk variance affects the course of kidney disease, with individuals carrying 2 APOL1 risk variants are more likely to progress to end-stage kidney disease. And this fact is variable, as you see on panel B. Patients with proteinuria kidney disease, shown in the red line, are more likely to progress to end-stage kidney disease or progress faster to higher grades of kidney disease than individuals who do not have proteinuria at baseline. So it is both the presence of proteinuria and presence of APOL1 high-risk genotype that predict the future course of kidney disease. Next slide, please. What about impact of APOL1 genotype on progression of kidney disease? In the CRIC cohort, the CRIC cohort consisted of both white individuals as well as Black individuals. And here, some of the individuals had diabetes, whereas in AST study, none of the patients had diabetes. So panel C here shows you impact of APOL1 genotype in patients with diabetes. And again, white individuals are shown in the light line. The middle line shows Black individuals with low-risk genotype, and the dark line shows you Black individuals with high-risk genotype. And what you see here is individuals carrying APOL1 high-risk genotype had the worst kidney outcome. And this remains true for patients without diabetes as well. Next slide, please. And also looking at, so not only presence of APOL1 risk alleles portends a poor course of kidney disease, it also portends poor response to traditional treatment. The mainstay for treatment for preventive kidney disease is better control of blood pressure and use of ACE inhibitors. What these graphs shows you is that better blood pressure control, a good tight blood pressure control, and use of ACE inhibitor were not able to alter course of kidney disease among individuals carrying APOL1 risk variants and had APOL1-mediated kidney disease. So we really need new treatments to manage patients who are carrying 2 APOL1 risk variants and have kidney disease. Next slide, please. So jumping ahead, and let's talk about kidney transplant, because I think this is a very fascinating natural experiment. Kidney transplants allows us to understand pathophysiology of APOL1-mediated kidney disease. APOL1 gene and APOL1 is present in all cells. However, the circulating APOL1 is predominantly made, is 100% made by the liver. And however, as I said, most of the cell types produce APOL1 protein, particularly locally, the kidney, right? So in individuals who are seeking kidney transplant, their own native kidneys are not working. As a result, there is no native kidney production of APOL1 protein. However, the native livers continue to produce APOL1 protein. So as a result, the kidney transplant allows us to conduct this natural experiment to understand, is it the circulating APOL1 produced by the liver, which is intact in patients on dialysis, or is it the local APOL1 protein that is produced by the new kidney that causes disease? If it is indeed the local APOL1 expression, then only the donor genotype, the new kidney donor genotype, will affect kidney graft outcomes. However, if it is the circulating APOL1 protein that affect outcomes, then only recipient APOL1 genotype will influence the graft outcomes, as the liver of the recipient will continue producing APOL1 protein, or perhaps it is both local and circulating APOL1 that causes kidney disease. Next slide, please. So let's first look at what is the role of donor APOL1 genotype, in this case, deceased donor, kidney donor genotype on graft outcomes. So in 2011, Dr. Barry Friedman and his group conducted this small retrospective study at a single center in Wake Forest. He enrolled 136 recipients who received a kidney from Black deceased donors. These deceased donors were genotyped for APOL1 high risk. As you see, 114 of this 136 recipients received a kidney from low risk, from Black donors carrying 0 or 1 APOL1 risk variance, shown in the dark blue line. 22 recipients received a kidney from Black deceased donors carrying 2 APOL1 risk variants. The graph here shows proportion with graft survival. You know, as you see here, on the lower the graft survival, more the graft will dip down. And what you see here is individuals carrying 2 APOL1 risk variants had a lower or inferior graft survival, as opposed to recipients who received kidneys from Black deceased donors with 0 or 1 risk variants. And as you see, the hazard ratio of graft loss was almost threefold. At around 2 years, a third of the graft losses occurred among the individuals who received kidneys from deceased donors carrying 2 risk variants. Of note, out of the 8 out of the 22 who experienced graft loss, 6 had FSGS-like lesions on their transplant biopsies. So next slide, please. Given the findings of this small study, this group of investigators expanded their study to include many more kidney transplant recipients across many more transplant centers. So in 2016, the authors reported their findings enrolling over 1,000 recipients, over 100 transplant centers, and they genotyped again deceased donors, and they looked at graft outcomes. Again, the recipient outcomes from deceased donors carrying 0 or 1 risk variants are shown in the black line in the graph seen on your right-hand side. And the outcomes from deceased donor carrying 2 renal risk variants is represented in the blue line. And what you see is recipients receiving kidneys from donors carrying 2 risk variants had inferior outcomes than the other group. Again, this study confirmed the findings of the smaller studies, suggesting that kidney donors, Black kidney donors carrying 2 APOL1 high-risk alleles conferred increased risk of graft loss. However, I want to point out again that 73% of the grafts from high-risk donors were working at 5 years. So similar to what I proposed in the native kidneys, that not all kidneys that carry 2 risk variants succumb to kidney failure. Majority of them are working well, even in terms of kidney transplant. Next slide, please. So far, I showed you the impact of donor genotype on graft outcomes. Let's look at APOL1 genotype of the kidney recipient, if that matters, because that would suggest that it is the circulating APOL1 produced by the liver that also affects the graft outcomes. So this study was published, I believe, in 2012 by a group in Boston, and they also had investigators from UAB. They looked at cohort of 206 kidney transplant recipients. Now, in this case, the recipients were genotyped for APOL1 risk variants. The donor were not genotyped, and these kidney transplant recipients were grouped based on the recipient genotypes. Because this group is enriched in Black patients with kidney disease, 50% of the cohort carried 2 risk variants. Individuals with 2 risk variants are shown in the dark, solid black lines on the left-hand side. Individuals carrying 0 or 1 risk variants are shown in the dashed line in the figure on the left. And what in the figure on the left depicts the allograft survival. And what you see here is there is no difference in graft survival based on the recipient APOL1 status. So for a long time, it was thought that it is only the deceased donor APOL1 genotype that predicts graft outcomes after kidney transplantation, and the recipient genotype did not matter, suggesting it is the local APOL1 protein produced by the kidney that affects graft outcomes. However, a recent paper, next slide please, published in Journal of Clinical Investigation just last year showed the contrary. This group genotyped around 300 recipients and donors enrolled in the GOCAR studies for APOL1 risk variants. And what you see here on the left-hand side is 3 groups based on the recipient genotypes for APOL1. The high-risk variants are shown in the blue line. The individuals carrying 1 risk alleles are shown in green line, and individuals carrying 0 risk variants are shown in the red line. And this graph shows you the likelihood of graft survival. What you see here is the number of recipient APOL1 risk variants was associated with increased risk of graft loss, independent of the APOL1 genotype. So these authors suggested that the APOL1 genotype of the recipient also mattered in predicting kidney graft survival. Next slide, please. The authors tried to look at what was the second hit that caused some kidneys with 2 APOL1 high-risk variants in the recipient, causing premature graft loss. And what they noted is that it was the increased risk of rejection. What you see is risk of rejection in recipients carrying 0 APOL1 risk variants was as low as 9%. However, recipients with 2 risk variants had rejection rates of around 45%. So perhaps it is the acute rejection that is the second hit that causes premature graft failure in selected individuals carrying 2 risk variants. So the jury is still out there as to whether the recipient genotype also confers inferior graft survival after kidney transplantation. Next slide, please. Now let's look at the last thing, which is, does the APOL1 genotype of the living donor matter? Now these are healthy individuals who want to donate a kidney to their loved ones so that they can get off dialysis. So to give you some background on living donors and their outcomes, let me review some landmark papers. And one of the landmark papers I would like to share with you was published in JAMA by Mazzali et al., where they looked at risk of developing end-stage kidney disease in living donors and compared that to healthy non-donors who were screened for good health at baseline. So these graphs shows you, likelihood of developing end-stage kidney disease in Black living donors. Middle panel is Hispanic living donors and white living donors. The blue line shows ESRD development in living donors. The black solid line is in non-donors selected for good health at baseline. And what you see here is not only the Black living donors are at the highest risk for developing end-stage kidney disease, but the absolute risk, that is the risk difference between the living donors and the non-donors, highlighted in this red parenthesis, is highest among the Black donors than the white donors. But I do want the audience to be mindful that the risk of end-stage kidney disease in living donors is far lower than that seen in general population. While the risk is higher than the healthy non-donors, it is far lower than that seen in general population. Next slide, please. Then these investigators went to look if it was the donor-recipient relationship that predisposed certain living donors to develop end-stage kidney disease. What this graph shows you that the biologically related living donors are more likely to develop end-stage kidney disease than biologically unrelated. So this begs the question that is it due to the shared genes between the donors and the recipients that predisposes certain donors to develop end-stage kidney disease? Next slide, please. Intrigued by the reports by Muzzali et al., myself and Dr. Goggins from Henry Ford Hospital conducted this small study of 136 Black living donors who had donated at 2 transplant centers in Michigan at Wayne State University and at Henry Ford Transplant Center, and we genotyped these donors for APOL1. There were 19 living donors who carried 2 APOL1 risk variants, and there were remaining 117 donors carried 0 and 1 APOL1 risk variants. So the prevalence of APOL1 risk variants in living donors was around 14%, similar to that seen in general population. Next slide. What we did then is looked at the post-donation renal function in living donors stratified by APOL1 risk variants. So if we look at the left-hand panel, figure 1, the figures in the right show the GFR at pre-donation and post-donation in the low-risk group, and the GFR for the high-risk group is shown in the black bars. What we see is individuals, living donors with the low-risk genotype had higher pre-donation GFR, and as a result had higher post-donation GFR after living donor nephrectomy. However, I would like to point out 2 kidney donors carrying APOL1 high-risk variants developed kidney disease, end-stage kidney disease in our cohort. So the next question we had is, does the APOL1 genotype affect the future trajectory of renal function after donation? To answer that question, let's focus on figure 2 on the right-hand side. What you see is GFR trajectory after donation, at least 10 years after donation, between the low-risk APOL1 genotype showed in the red line and the high-risk genotype shown in the black line. And what we see is individuals carrying high-risk genotype had a faster decline in their kidney function after donation. In the background, you see several faint lines, and what those depict is trajectory of individual donors. What I want to highlight to the group is there were some donors in the high-risk category that had very accelerated decline in their kidney function, and there were quite a few of them who had pretty stable kidney function post-donation in the high-risk genotype. Again, emphasizing the fact that not every live kidney donor who carries APOL1 high-risk variant is going to develop end-stage kidney disease or develop kidney failure down the road. Only select individuals develop kidney disease, similar to what we have seen with general population in native kidneys, similar to what we have seen even in kidney graft outcomes. Next slide. So then that begs the next question, does the act of donation, mere act of donation, affect the trajectory of kidney function in individuals stratified by APOL1 genotype? So to answer this question, we compared the renal trajectory of healthy non-donors from cardiac study who were selected for good health at baseline. We matched these non-donors based on age, gender, and race to donors and their APOL1 genotype and family history of end-stage kidney disease. And what we see here is the trajectory of renal function for individuals with low risk APOL1 genotype in the left panel in panel A, and the panel B shows you trajectory of renal function for the high risk genotype. What we see is the act of donation did not adversely affect the interact with APOL1 genotype and renal function at follow-up. Next slide, please. So we concluded that APOL1 high-risk genotype in Black living donors is associated with lower pre and post-donation eGFR, and also some of the individuals with this high-risk genotype experience faster decline after donation. Note again, not all donors with high-risk genotype had accelerated decline in kidney function. While high-risk APOL1 status did not modify the association between donation and rate of decline in GFR, individuals with high-risk genotype who have lower GFR after donation may experience a faster decline in GFR and therefore may reach end-stage kidney disease sooner. Next slide, please. So given the widespread availability of APOL1 genetic testing and still uncertainty around the utility of APOL1 testing and whether a living donor who's carrying 2 risk variants should be not allowed to donate, so we want to protect a donor on one hand, and at the same time, we know that the Black individuals with kidney disease have a lower access to living donor transplant. We wanted to balance their access to living donation, and therefore we created a group of, had a work group of experts who came up with the guidelines as to how to best integrate APOL1 kidney risk variant testing in context of living donation. Again, this is just the opinion of the panel and consisted of a transplant nephrologist, an ethicist, a genetic specialist, a surgeon, and a patient. It is very important to have patient voice also while we create these guidelines. Next slide, please. So the consensus was that every patient who reports some African ancestry, particularly coming from recent African ancestry from the sub-Saharan region, should be educated, should be told about the APOL1 gene and its association with kidney disease. What the work group insisted is that the provider should be mindful of that there is a lot of individuals who are of mixed race, who come from Brazil and other areas, that we should be asking about the African ancestry and talk about APOL1-mediated kidney disease. The physicians or providers should not look at facial characteristics to determine who should and should not be educated about APOL1 kidney risk variants and its association with kidney disease. So again, as I mentioned, shown here, individuals, Africans from Africa, Africans from North America, Afro-Caribbeans, and individuals from Africans from Brazil should be educated about APOL1-mediated kidney disease. Next slide, please. Again, as I said, providers should ask potential donors about their ancestry and ethnic identity rather than rely on their own perception or medical records. And the reason I say so is because there was a paper published in CJSON where the patients want to know about APOL1 and its association with kidney disease and all the uncertainties that we have today about the exact impact of APOL1 on future risk of development of kidney disease. So I think it is better to be very transparent and talk to our patients in clinic about APOL1 gene. Next slide, please. With regards to living donation, who should be tested? This graph on the left shows you interaction of race and age on future risk of end-stage kidney disease. The risk of end-stage kidney disease is highest in individuals shown in the dashed line, which are the Black individuals. And as you see, the risk varies based on age. Young individuals, particularly age 45 and under, are at the highest risk of developing end-stage kidney disease. Therefore, the work group proposed that Black living donor candidates age 45 and under who are related to their first-degree relatives to their intended recipients should be educated about APOL1 risk variance and its association with kidney disease, and perhaps be tested based on other risk factors for kidney disease, such as history of smoking, borderline low GFR at the time of donation, or if they were obese or carried extra weight. Next slide, please. So let's kick off with some clinical vignettes that you may face in your clinic. So I would like to jump off to case number 1. Next slide. Sorry, before that, before jumping off to the case presentation, I just want us all to be mindful before we order APOL1 genotyping. Our obligation is to be as transparent as possible and give all the possible known information to date, both about the known information about APOL1 gene and its possible association with kidney disease, and the unknowns about its absolute association, lack of available treatment, and how it can refine the risk of progression of kidney disease and improve risk stratification, particularly when evaluating a living donor candidate. We should also acknowledge the downstream effect and be mindful as we order tests on our patients seen in clinic. It may result in some anxiety around the test results, and particularly in lack of available treatment. It may further stigmatize the community and further restrict the pool of living donor for Black patients on dialysis. Next slide, please. So with that said, let's jump to case number 1. I happen to see this 40-year-old Black male with biopsy-proven FSGS in your clinic for treatment. I would like to ask the group, would you like to perform APOL1 genetic testing? Yes or no? And I'll give you a minute to answer. Okay, so I think for now, as we unwrap, so I see most of you said yes, and I would agree to that. While a lot is unknown, at least we can talk. Can you please close this poll? Thank you. So I would too, perform APOL1 genetic testing to inform the patient, and perhaps he can even inform his loved ones, so they all can be mindful about kidney disease and address the risk factors, such as be more careful with their diet, with their salt intake, better control blood pressure, and better follow up with primary care physicians. Hopefully there will be targeted treatment for APOL1 associated kidney disease, particularly folks with FSGS and those who have proteinuria kidney disease. And as mentioned before, Vertex is going to be launching that trial. So I think I would agree with the group and go ahead and perform APOL1 testing. So the next question I have is, if he tests positive for APOL1 high-risk genotype, he asked you, are there any approved treatments? And the answer right now is no, but you could tell your patients that there are treatments, possible treatments underway, and if there is not one today, he may be eligible for one down the road. Next question, please. And I want to just show a brief outline on the treatment paradigm. As I said, that currently we don't have any FDA approved treatment, but we have, there are a lot of treatments who are, that are being developed underway, such as CRISPR treatment that targets the APOL1 gene itself. There is treatment with antisense RNA that attacks the mRNA transcripts for APOL1 gene. That is the messenger RNA that produces the APOL1 protein. And Vertex right now is launching a trial against the APOL1 protein. So there are a lot of trials that are going to come down the road. So stay attuned, and hopefully your patient, may be eligible for one of those trials down the road. Next slide, please. So now this very young individual has reached dialysis, and he's now seen in transplant clinic. And, in transplant, when we see an individual with FSGS, we have to counsel them that there is a risk of recurrence. The number we give out is 30% of individuals with FSGS are likely to develop recurrent FSGS in the renal allograft. Now, the fact that this gentleman is carrying 2 APOL1 risk variants, he's asking, hey doc, what are my risks for recurrence of FSGS in my allograft after kidney transplant? What would your answer be? Is it 0 risk of recurrence? Same as before, 30%, or we don't know? So I see there is a split between 30% risk of recurrence and don't know. I would say, I don't know. More to come. As I showed you before, we don't know whether it is the donor genotype that matters or whether it's the recipient genotype that matters that causes recurrent FSGS. So more to come. I think in the interest of time, I'm going to skip the next, maybe next 2 slides, next 2 cases, as I see we are running short of time. So what is the role of genetic counselors in APOL1 screening? Next slide, please. So counseling can be performed by transplant nephrologist or a member of a transplant team, or even by a genetic counselor, if you have access to one. But I want to strongly emphasize that counseling should be offered before genetic testing. We need to inform the patient at hand about all the knowns and unknowns associated with APOL1 genetic testing. And if the patient still wants to know the genetic test results, then only move forward with APOL1 testing. Because I showed you that while there are some benefits, there are no treatment, and it may create some anxiety and angst for the patient, for the rest of the family members. And, and there could be some stigmatization. There could be some denial of insurance, particularly disability insurance. So one should be cognizant before ordering genetic testing, and definitely perform counseling before and after genetic testing, and review both the pros and the cons of testing. Acknowledge the lack of robust data that not every individual carrying 2 risk variants is going to get kidney disease. Only 20% or so develop kidney disease. And there is a second hit that causes kidney disease. So mere carrying 2 APOL1 risk variants is not going to result in kidney disease. And also acknowledge currently there is lack of effective treatment. Next slide, please. Where can I find a genetic counselor if you don't have one at your institution? The National Society of Genetic Counselor is an excellent resource for learning more about the role of genetic counselor in all renal genetic panels, including APOL1. And please refer to this database that tells you availability of genetic counselors by state. Next slide, please. Where can I send my patients to learn more about APOL1 and APOL1-mediated kidney disease? Next slide, please. The American Kidney Fund has a new patient and caregiver education campaign about APOL1. Assets include a patient-friendly website, an archived patient-centered webinar, guides for talking to healthcare team, and more. Please visit their website for more information. Future directions. I already alluded to the slide on, the treatments, possibly, treatments in the pipeline. There are several NIH-sponsored studies underway, mainly the APOLO1. Next slide that I would like to discuss. The APOLO is a NIH-sponsored study that consists of 260 transplant centers, 58 OPOs, 6 HLA labs that plans to enroll every single Black kidney donor in the country, both living donors and deceased donor, and also wants to enroll every single recipient from the Black donor in the study, both donors and living and deceased donors, and the recipients of the kidneys are going to be genotyped for APOL1 risk variants. And then we're going to follow them for at least 3 to 5 years to see the impact, whether it is the donor genotype or whether it is a recipient genotype that portends graft outcomes. And I see Dr. Gee in the audience, Patrick Gee, and who is also part of this study. So this is a very exciting study, and we have a patient advocate group guiding us through the study. There are several ancillary studies that have spinned off this Apollo study, one looking at the outcomes in living donors, one looking at ethics and how do you return genetic testing results, and many more. So a lot more to come. This is a very exciting field. Next slide, please. So I would like to end this talk with some take-home message. At present, there is no utility in performing APOL1 genetic testing in healthy Black individuals, except in context of living donation or if they have proteinuria kidney disease. I want to remind all of you, not all individuals carrying 2 copies of APOL1 kidney risk variants are going to develop kidney disease. A second hit is required. Black individuals with proteinuric kidney disease are likely to benefit from some such screening. Genetic counselors play a key role in educating patients both before, I want to emphasize counseling before and after testing. Therapeutic trials in native kidneys are ongoing and hopefully may start soon in kidney transplantation too. Thank you so much for your time and attention, and happy to take any questions.
Mona, thank you. That was a terrific talk. A lot of great information in there. While we're waiting for questions from the audience, I wanted to ask you 2 things, if I might. You made some comments about diabetic kidney disease, and I know that often we're talking about APOL1 in the context of non-diabetic kidney disease. So 2 questions. It looks like there was an additive effect of high-risk APOL1 variants on progression of diabetic kidney disease. And was this just additive, or does there seem to be some synergy between diabetic kidney injury and APOL1? And the second part of the question I just wanted to ask you is, does that mean then we should think about in our Black patients with diabetic nephropathy, should we also be testing them for APOL1?
Great question, Silas. I think as this field is evolving and we're learning more, what I wanted to remind you is of the background slide where we had a blue cone showing maximal impact of APOL1 with HIV nephropathy, then less so in hypertensive associated ESRD. So it looks like there is some impact of APOL1 genotype, high-risk genotype in progression of kidney disease in the diabetics, but the impact is not as great as one would see in plain garden variety of hypertensive associated kidney disease, if I may.
Perfect, terrific. And probably just backing up a little bit from the donors a little bit, just in the general population. And so are there recommendations now for otherwise asymptomatic Black individuals in the population who have a family history of kidney disease in terms of whether or not those folks should be tested for APOL1?
I think population-based studies have failed to show such a significant association between APOL1 risk variants and kidney disease. So at the present time, they are discouraging population-based screening. Now, the question you asked me, if you have a family member with kidney disease, and if you're totally asymptomatic, should you be tested? I think at the present time, I would just say no, because my guidance right now, what would we tell a patient in clinic? Make sure your blood pressure is better controlled, right? Make sure you don't develop diabetes, make sure you watch your salt intake, make sure you lead a active, healthy lifestyle, which you would tell a patient who has a family history of kidney disease. And the APOL1 is not going to change those recommendations at the present time. So I would not suggest that we start screening family members who are asymptomatic for APOL1 high-risk genotype, unless they have manifested disease such as with proteinuria.
Perfect, perfect. Thank you. And we have a question from the audience, which I think a lot of people have this question, which is thinking about the cost. So the cost of the APOL1 genotype testing, whether there's potentially financial assistance for this testing. And then I wonder if you could just comment on the availability of the testing as well.
Yes, thank you. Great question. So I think, so the cost is around $300 to $400 based on the lab. And, as more and more folks use it, perhaps the cost should drive down, right? And there are several private labs that conduct APOL1 testing. Most of them, I don't think so. So this is a send-out test for most, even large universities. It is not an in-house test. And these companies offer genetic counselors for those who are unaware of such assistance, right? They offer genetic counselors who could talk to the patient before and after test results too. And with regards to insurance coverage, again, I think it depends on the type of the insurance, the context in which the testing is done, right? So if you have someone, who's in a transplant clinic, who you need to do to prognosticate, the transplant center generally covers it. Or if you have a healthy living donor and you want to test them for further risk stratification, then the transplant center covers that cost. But in CKD clinic, if they have proteinuria kidney disease, then my guess is the insurance company should cover that. This is part of, regular workup.
Perfect, perfect.
And again, this is part of more and more of personalized medicine, if I will, right? Just because you're Black doesn't mean you only can get APOL1-mediated kidney disease. If you have proteinuria kidney disease, you could have, actin, portosin, a lot of, mutations for structural, diseases that affect the podocytes, right? So I think what we see in clinic is, if you have an individual with proteinuria kidney disease, you biopsy them, it shows FSGS, then you send out a genetic panel. And one of the items is APOL1, in addition to several other markers.
Perfect. So no, that's super helpful. And I wonder, I think we have time for one other question, which I think follows nicely from your last answer, which is, in addition to the APOL1 genotype testing, kidney biopsy, other diagnostics, can you combine other testing with APOL1 testing to give you prognostic information?
Yes, I think kidney biopsy is always useful because it tells you at the tissue level what is going on in the kidney, how much fibrosis there, how many filters, if you will, how many glomeruli are affected. So APOL1 is just a piece of the puzzle. I think you get a lot more information, not only with the kidney biopsy, but with the simple, cheap test of urine analysis and how much protein is there in the urine, right? The greater the degree of proteinuria, the more likely the patient is going to progress quicker into kidney failure.
Perfect. Terrific. Well, I thought that was great. That was great. I really appreciate you taking the time to join us. So as we close, I just want to take a moment to thank Dr. Doshi again for just a terrific talk, a lot of great information. I want to thank Vertex for their support, and I want to thank each of our participants for joining us this afternoon. And remember, there's some great information for providers and for patients on the American Kidney Fund website.
Thank you, Silas, and thank you, AKF, and see you all. Have a good afternoon.
Good afternoon, everyone.